Hello, this presentation on DNA structure and function is intended for students taking Anatomy and Physiology 1 at Madison College with Gina Piscatelli. Please refer to your course syllabus for the appropriate reading assignment and study questions. The helix shown on this slide is most likely recognizable to you. The picture of the cell on the left shows that DNA is found in the nucleus. Its function is to provide a code of instructions for the amino acid sequence of polypeptides to be synthesized by the cell's ribosomes. Notice that the DNA is sequestered in the nucleus, but the ribosomes are located in the cytoplasm. Therefore, a copy of the DNA instructions in the form of RNA travels through the nuclear pores to the cytoplasm, where the ribosomes will synthesize proteins according to the DNA instructions. To understand the roles of DNA and RNA better, let's first review what you already know about their structures. Both DNA and RNA are chains of nucleotides linked together with phosphodiester bonds. The backbones of the DNA and RNA strands contain the 5-carbon sugars and phosphate groups of the nucleotides. On the left, we see double-stranded DNA, and the 5-carbon sugar is deoxyribose. And in single-stranded RNA on the right, the 5-carbon sugar is ribose. In DNA, the nitrogenous bases adenine, thymine, cytosine, and guanine point towards the interior of the molecule because it's double-stranded. The two strands of DNA are linked together with hydrogen bonds between the bases. The bonding between bases is complementary, in which adenine only binds to thymine and cytosine only binds to guanine. In RNA, the nitrogenous bases are the same, except that uracil replaces thymine. Due to the unique structures and locations of DNA and RNA, they perform different roles. DNA contains the entire genetic code, whereas RNA will be a short segment of nucleotides that is a copy of a gene. DNA remains in the nucleus where it is protected, but RNA is made in the nucleus and then moves to the cytoplasm where ribosomes can read the code. DNA needs to remain intact for it to maintain its integrity, and it provides the genetic code that will be inherited. But RNA is made each time a protein needs to be made. Before we get into too much detail, let's look at the big picture concerning the role of DNA, its genes, and the inheritance of traits. DNA dictates our traits by coding for proteins. Some proteins will provide structure to a cell, a tissue, maybe an organ, and ultimately our bodies. Some proteins instead contribute to a visible characteristic, such as eye color or hair color. Other proteins are enzymes. These enzymes catalyze the production of other types of organic molecules, such as carbohydrates and lipids. For example, shown here, DNA would code for the enzyme lactase, which transforms lactose into glucose and galactose. This slide illustrates how a gene can code for a visible trait. The human genome consists of 46 chromosomes, but in this picture we see one. It's purple, and its regions that are colored blue and pink are particular genes. One gene is shown blue and that codes for a protein that contributes to an eye pigment or eye color, whereas the pink gene codes for a protein or a pigment that contributes to hair color. What makes these two genes distinct is that they each have a different sequence of nitrogenous bases. These nitrogenous base sequences are shown in the lower box, and you see that the eye color gene starts with ACCGT, whereas the hair color gene starts with TTTCC. If we look more closely, we see that the genetic code in DNA is a sequence of nucleotide bases that dictates the amino acid sequence of a protein. In fact, 3 DNA nucleotide bases ultimately code for one amino acid. For example, CCT codes for the amino acid glycine, and GTA codes for the amino acid histidine. In this manner, the particular order of nucleotide bases in DNA determines the primary structure of a protein. A chain of particular amino acids determines this primary structure. With our understanding of DNA and RNA, let's look at the specific process of protein synthesis. There are 3 steps. The first 2, transcription and RNA processing, occur in the nucleus. The third step, called translation, occurs in the cytoplasm. During transcription, this first step, a complementary RNA copy of the gene is made. This RNA copy is called the primary transcript. During the second step, called RNA processing, the primary transcript is modified. The end product of this step is called messenger RNA, or mRNA for short. Before the third step can begin, messenger RNA leaves the nucleus and enters the cell's cytoplasm. During the third step, ribosomes in the cytoplasm attach to the messenger RNA, read the nucleotide sequence, and assemble the protein. These ribosomes make sure that the correct amino acids are joined together with peptide bonds. The terms transcription and translation were derived from the fact that transcription means the copy is written in the same language as the original, that is, nucleotides. Translation means the copy is written in a different language than the original. In our situation, the final step, translation, starts with a nucleotide sequence in RNA, and the final protein is in a sequence of amino acids, a different language. So the ribosomes perform translation. Let's look more closely at how the genetic code is first transcribed, and then it's translated into a sequence of amino acids. First, when transcription occurs, the RNA copy is synthesized with a sequence of bases that are complementary to the sequence of bases in the DNA. This is because the RNA strand will temporarily bind to the DNA strand. In the diagram on the slide, notice that the first 3 DNA nucleotides are TAC. The copy that is complementary to that would be ATG if we were to write it in DNA. But remember, we're using RNA, which doesn't have thymine. It's replaced with uracil. So instead, the RNA copy will be AUG. Secondly, when translation occurs, 3 nucleotide bases provide the code for one amino acid. These 3 RNA bases are called a codon. The amino acid encoded by the codon AUG is methionine. It is known which amino acid is coded for by each codon, and this table is used to predict the amino acid sequence that will be made from the primary transcript of sequence of codons. To use this table, you need to know the messenger RNA sequence. It's written in codons. The first base is listed in the first column. The second base is listed in the second, third, and fourth columns horizontally on the table. And the third letter is listed in the last or the fifth column. So in the previous slide, the start codon AUG, boxed in red on this slide, codes for the amino acid methionine. There is an enzyme in the nucleus that performs gene transcription and makes the RNA primary transcript. Its name is RNA polymerase because it's making a polymer with many subunits. To begin, the RNA polymerase binds to a region on the DNA called a promoter. Every gene region has an upstream promoter to which RNA polymerase binds. This diagram shows the promoter region in yellow. The purple RNA polymerase attaches to the promoter. RNA polymerase begins moving along the chromosome from the promoter region, unwinding the DNA as it moves, and it reads the nucleotide base sequence. Once the RNA polymerase encounters a sequence called the start sequence, it will begin to make the RNA molecule. The DNA start sequence is TAC. The complementary RNA sequence that will be assembled by RNA polymerase is AUG. RNA polymerase continues to move along the chromosome and synthesizes a complementary strand of RNA until it reaches a stop sequence. At this point, the DNA rewinds, the RNA detaches, and the RNA polymerase falls off. So in summary, the first part of protein synthesis is called transcription. Transcription is performed by the enzyme RNA polymerase. RNA polymerase synthesizes a primary transcript that is in an RNA sequence complementary to the original DNA gene sequence. You may notice that many diagrams such as this one depict a visual break every 3 bases. That is because 3 nucleotide bases code for 1 amino acid. The next step of protein synthesis involves modifying the primary transcript before the RNA can leave the nucleus. The ends of the messenger RNA or the RNA will be capped with certain molecules. That enables the RNA to leave the nucleus and not be degraded. Another process that occurs during RNA processing is called splicing. Splicing refers to the removal of nonsense or non-coding regions. The regions that are spliced out are called introns. They are extra DNA nucleotides that act as transcription regulators. They are not destined to become part of the functional protein. After introns are removed, the remaining codons, called exons, are joined together. Once the exons are joined, the RNA is mature and it is referred to as messenger RNA. After RNA processing has occurred, messenger RNA leaves the nucleus through nuclear pores to enter the cytoplasm. Once the mRNA is in the cytoplasm, ribosomes will attach to it and begin the process of translation. This leads us to the last step of protein synthesis called translation. During this process, The ribosome reads the sequence of messenger RNA bases and synthesizes the appropriate protein. This process is called translation, remember, because the molecular language changes. Messenger RNA is written in nucleotides, whereas the protein will be written or consist of amino acids. In addition to messenger RNA, there are other types of RNA involved in translation. One type, called ribosomal RNA, is a component of the ribosome, and it helps to keep the messenger RNA stabilized within the ribosome. The other type, called transfer RNA, will bring amino acids to the ribosome. There are many transfer RNAs, one for each mRNA codon. Each transfer RNA has an anticodon that is complementary to a codon, and that transfer RNA is attached to a single amino acid. This diagram might help you understand transfer RNA better. Notice the transfer RNA molecule in the black circle. The brown squiggly line represents a sequence of RNA nucleotides. Notice in the flat region there are 3 nucleotide bases shown as UUU. This is the anticodon, and it is complementary to a messenger RNA codon of AAA. Also notice on the transfer RNA that one amino acid, lysine, is attached. Lysine will always be on a transfer RNA that has this particular anticodon of UUU. That is because the codon for lysine in messenger RNA is AAA. So the transfer RNA needs to be complementary to that. So the process of translation is initiated when a ribosome binds to the messenger RNA. In this diagram, the ribosome is shown as 2 parts, yellow and blue. As the ribosome reads or scans the messenger RNA, it looks for a start codon. AUG is the start codon. The transfer RNA with a complementary anticodon, UAC, will then bind to the messenger RNA. The transfer RNA molecules bring in the appropriate amino acid. 2 important things about transfer RNA molecules again. Only a transfer RNA with the correct anticodon will fit into the ribosome, where the messenger RNA codon is. As multiple transfer RNAs arrive, bringing sequential amino acids, peptide bonds are formed between the amino acids. So on the diagram there will be a peptide bond between methionine and phenylalanine, and another one eventually between phenylalanine and lysine. In this way, a polypeptide will continue to grow. This slide shows a peptide bond between 2 amino acids that were brought to the ribosome by transfer RNA. As the ribosome continues to move along the messenger RNA, the process of moving is called translocation, and that continues until a ribosome reaches a stop codon. This stop codon does not have a complementary transfer RNA, so no new amino acid can be added to the growing polypeptide chain at this point. When this occurs, the translation machinery will disassemble. The ribosome will release 3 things: the messenger RNA, which will just be digested, the transfer RNAs, which can be reused, and the polypeptide. So to summarize, the process of translation involves a starting molecule, messenger RNA, and then the final product is a polypeptide or a protein. Ribosomes read the messenger RNA codons. The amino acids are brought to the ribosome by transfer RNAs. The ribosome ensures that the correct transfer RNA anticodons bind to the messenger RNA codons. This ultimately ensures that the protein has the correct sequence of amino acids. As transfer RNAs arrive with new amino acids, these amino acids are linked together with peptide bonds. This bond formation is catalyzed by the ribosome. We will perform an exercise in class in which you are given a gene sequence as shown in the top box, TAC, CGC, CCT, TGC, GTA, CTC, ACT, crazy, huh? You should be able to predict the sequence of a primary transcript for this gene sequence. It will contain complementary RNA bases. And as shown here, A is complementary to DNA T, U is complementary to DNA A, etc. I will never ask you to know which are introns or exons, and you won't have to splice anything out. Using an mRNA sequence and the table we looked at earlier, you should then be able to determine the polypeptide amino acid sequence that's coded for by the messenger RNA codons. In this case, the amino acid sequences methionine, alanine, glycine, threonine, histidine, and glucine. Notice that you don't even have to know anything about the transfer RNA anticodons to make this prediction. But it is interesting to note that the sequence of anticodons on the transfer RNAs, which are carrying the amino acids, is identical to the original DNA sequence. Except, of course, all thymine is replaced with uracil. Thank you very much, and I'll see you in class with an exercise for this.